Okay, part 3 of photosynthesis, the Calvin cycle. Well, part 3 of this lecture, part 2 of photosynthesis, the Calvin cycle. So the Calvin cycle, like the citric acid cycle, is a cycle. So it regenerates the starting material after molecules enter and leave the cell. So when a molecule enters, it adds on to a starting material, and then it makes what's going to start the next one. So yeah, it regenerates its starting material after molecules enter and leave the cycle. The Calvin cycle is anabolic. So anabolic means building things. So it builds sugar from smaller molecules by using the ATP and the NADPH that come from the light reaction. So the ATP puts in the energy, and then the NADPH brings some reducing power of electrons that are carried on it. So carbon enters the cycle as carbon dioxide. So the plants breathe in carbon dioxide. And then the carbon leaves from one of these guys through or as a sugar called glyceraldehyde 3-phosphate, or G3P, and that could be used to make glucose really easily. So that's what we're actually tracking. So for the net synthesis of one molecule of G3P, this cycle has to take place 3 times. And then each time it's making it's adding on a carbon. So one carbon from one molecule of carbon dioxide gets put on, then another, and then so G3P has 3 carbons, and they come from those molecules of carbon dioxide. And so there are 3 phases of the Calvin cycle: carbon fixation, reduction, and regeneration. Yeah. Oops. So the carbon fixation, like it's putting it somewhere. Yeah, we'll go through each one of these. So carbon fixation, reduction, and then regeneration of the acceptor. So carbon fixation, phase 1, the binding of carbon dioxide to a 5-carbon sugar that's already around named ribulose biphosphate, or RuBP, is catalyzed by this enzyme that's called RuBP carboxylase oxygenase, or Rubisco. So the molecule that you're starting with, and then you add the carbon from, is RuBP. And then the enzyme that does it is called Rubisco. So that next intermediate goes from having 5 carbons and then another carbon added on to now being a 6-carbon intermediate. And then this molecule, in this intermediate molecule, is immediately split into 2 molecules of 3-phosphoglycerate. So phase 2, reduction. Each molecule of 3-phosphoglycerate is altered through phosphorylation by 6 ATP and reduction by 6 NADPH to ultimately produce a G3P sugar. So for every 3 molecules that enter the cycle, 6 molecules of G3P are formed, but only one of these can be counted as a net gain of carbohydrate. The remaining 5 molecules of G3P are rearranged in a complex series of reactions, yielding 3 molecules of that starting thing again, RuBP. So 3 additional molecules of ATP are used then to help facilitate that regeneration of RuBP. So here's showing that visually. So there are 3 carbons coming from 3 molecules of carbon dioxide. Each one gets put on. So Rubisco adds the carbon to RuBP. Now this is a 6 carbon intermediate. Then it gets chopped in half basically, and then so there's this 3 carbon molecule, 3 phosphoglycerate, and by using the energy from 6 molecules of ATP, it adds on a phosphate, and then so now you have 1,3 biphosphoglycerate. And then with the electrons and the reduction, you make G3P. So 1 molecule of G3P gets put out, like spit out basically, to make glucose and other organic compounds. And then the 5 other molecules of G3P get rearranged to remake RuBP so that the next cycle can start. So for the net synthesis of 1 G3P molecule, the Calvin cycle uses energy. Remember, it's anabolic, you're building a molecule. So you're using 9 ATP molecules and 6 molecules of NADPH. And so then, the light reactions help regenerate the ATP and NADPH that powers the Calvin cycle. And the G3P is the starting molecule for metabolic pathways that make other organic molecules, including glucose, sucrose, and other carbohydrates. So it's, we still track it with glucose because G3P can make glucose. And then it kind of keeps it more aligned with cellular respiration. So this is a big picture overview. So, when you water a plant and the plant is receiving sunlight, it's going to use those things, plus the carbon dioxide that it's breathing in through the stomata. It's going to use those to power these 2 processes that overall make photosynthesis happen. So in the light reactions, the light kind of bounces around. Here's all the different, where the energy is at this point in time. It goes from photosystem 2, an electron transport chain. Photosystem 1, an electron transport chain. It makes ATP and NADPH, and those help power the Calvin cycle. And through that, you're actually using the carbon dioxide to build sugars like glucose or other kinds of sugars like sucrose, or maybe if you wanted to store a bunch of sugar because winter's coming, so you'll store it in the form of starch, a long chain of glucose molecules. And then so the plants have to breathe out their oxygen and contribute to the continuing circle of life. And so some plants have to adapt a little bit differently. They have to adjust their photosynthesis. So dehydration is a major challenge of terrestrial life for plants. I'll leave it at that. So dehydration is a major challenge of terrestrial life for plants, so stuff that's on land. Especially in arid climates that are hot, right? You run the risk of dehydration. So plants that live in these environments have metabolic adaptations to help conserve water, but then these adaptations have trade-offs. So one important trade-off is the balance between photosynthesis and conserving water. So on hot, dry days, plants can close their stomata, those little pores that let that gas exchange happen. So this helps conserve water, but it also limits photosynthesis because the closing of the stomata reduces access to the carbon dioxide and then causes oxygen to build up. So it's kind of like if we just stopped breathing, we're going to build up a waste product, and we're not taking in other product that we need. So it's kind of a trade-off, right? So these conditions favor an apparently wasteful product or process called photorespiration. So this is a different process, but it's kind of wasteful. So most plants are what are called C3 plants. So that initial fixation of carbon dioxide via rubisco, that enzyme, it forms a 3-carbon compound. So that's why it's C3 plants, because at this point it forms a 3-carbon compound, the 3-phosphoglycerate. In photorespiration, rubisco binds with oxygen instead of carbon dioxide, and so this produces a 2-carbon compound. And photorespiration is costly because it consumes oxygen, organic fuel, without producing any ATP or sugar. So it's not going to really help. I mean, it's not going to power the Calvin cycle, and so then it's not going to make sugar. So it's costly. Like, it's consuming something and it's not really making anything. So it's thought to be an evolutionary relic because rubisco, that enzyme that works on RuBP and slaps on that carbon, it's thought to be an evolutionary relic because rubisco first evolved at a time when the atmosphere had a lot less oxygen and more carbon dioxide. So yeah, if we go way back in evolutionary time, our atmosphere didn't have as much oxygen as it has today. All that oxygen actually came from photosynthetic bacteria, cyanobacteria. So there was like a lot more carbon dioxide in the air. And then from cyanobacteria making oxygen through photosynthesis, after so much time, it built up oxygen in the atmosphere. So there had to be a different process, right? And so this is what we think this comes from. Photorespiration. So photorespiration can provide some protection from the damaging products of the light reactions that build up when the Calvin cycle slows due to low carbon dioxide. So for example, again, in us, if we were to hold our breath, like, we are building up waste product. In us, our waste product is carbon dioxide. It's like if we evolved a way to break down the carbon dioxide. So yeah, what's actually more, or the primarily like worst thing for you if you were to just hold in your breath for a long time is that buildup of carbon dioxide. So it's like if we were to evolve something to use that waste product and not have it build up. So in a way it's not great, but it's also not killing you. So it's wasteful, but yeah, it's keeping you alive basically. Yeah, so we think that this is why photorespiration is still around. It's like consuming fuel without making any ATP or sugar, but we think it's to kind of help them, stay alive and have less damaging effects of those light reactions products building up. So in many plants, photorespiration, when they turn to that, it drains as much as 50% of the carbon fixed by the Calvin cycle. And so in some plant species, there are different modes of carbon fixation that have evolved to help minimize photorespiration and optimize the Calvin cycle. Sorry, the lights in my eyes. So there are plants called C4 plants. So C4 plants minimize the cost of photorespiration by incorporating carbon dioxide into a 4-carbon compound as the first product of the Calvin cycle. So that's why it's called C4, because that first intermediate you're making has 4 carbons. And so this process in C4 has evolved several times and is used by several thousand species in at least 19 different families. So it's really interesting when that happens. Like, it's happening independently in these different things, and then it happened to happen. And so some important agricultural examples that have evolved to this include sugarcane and corn. So in hot, dry weather, C4 plants partially close their stomata. So this helps like conserve water, but also reduces carbon dioxide. In these plants, photosynthesis begins in mesophyll cells, but then gets completed in other cells called bundle sheath cells. These are cells that are arranged in tightly packed sheaths that are around the leaf veins. So it's like this process of photosynthesis starts here, but it actually gets finished here, somewhere else, by the leaf veins. And so sugar production in C4 plants occurs in a 3-step process. So step 1, the production of that 4-carbon precursor is catalyzed by an enzyme called PEP carboxylase inside the mesophyll cells. PEP carboxylase has a lower affinity for carbon dioxide than Nabisco does. So it can fix carbon dioxide even when carbon dioxide concentrations are low. Step 2, the 4-carbon compounds are exported to those other cells, the bundle sheath cells, through that connection between plant cells, plasmodesmata. It just like shoots it through. And then step 3, within those bundle sheath cells, carbon dioxide is released from that 4-carbon compound and then used in the Calvin cycle. Then pyruvate is transported to the mesophyll cells where 1 ATP is used to convert it back to PEP. And then this ATP is regenerated during cyclic electron flow. So here is a picture of that. Here is a stoma, so a singular stomata. And then here is a bundle sheath cell that surrounds the veins in the plants and leaves. And then here are the mesophyll cells. So then, because they're interconnected directly by those plasmodesmata, they can just make this, shove it into the bundle sheath cell, and then that's what's going to finish making the sugars here in the Calvin cycle. And then it makes pyruvate, which can then go back and help remake PEP. So this is a way to still make sugar. And in a super bummer, CO2 levels in the atmosphere have drastically increased because of human-caused changes to the carbon cycle. So carbon dioxide levels have drastically increased since the Industrial Revolution began in the 1800s, and they continue to rise today due to human activities. So this increasing carbon dioxide and temperature may affect plants differently, perhaps changing the relative abundance of these species. So the effects of such changes are unpredictable and are a cause for concern. We're messing up nature, basically. And suitable agricultural land is also decreasing because of climate change, while the world population and demand for food continue to increase. So we might end up depending a lot more on the C4 photosynthesis than C3, because C4 photosynthesis uses less water and resources than C3 photosynthesis. So scientists have genetically modified rice, which is normally a C3 plant, to carry out photosynthesis for an estimated 30 to 50% increase in yield for given water and resources. And then there's another way too. So some plants, including succulents, so if you have those as a house plant, they conserve water by using CAM or crassulacean acid metabolism to fix carbon. I just know it as CAM. So CAM plants open their stomata at night and incorporate CO2 into organic acids that are stored in the vacuoles. And then during the day, the stomata close, and then CO2 is released from the organic acids and used in the Calvin cycle. So it's kind of like a little time-dependent mechanism. And the CAM pathway is similar to the C4 pathway in that they both incorporate carbon dioxide into organic intermediates before it enters the Calvin cycle. And the C4 pathway structurally separates the initial steps of carbon fixation from the Calvin cycle. Yeah, they happen in different cells. In the CAM pathway, the steps occur in the same cell, but are separated in time. So this is a comparison of C4 and CAM. So normally in a plant, comparing C4 and CAM. So in C4, photosynthesis, this part happens in the mesophyll cells, so they're separated by different cells. It's carbon dioxide is put into an intermediate, and then it gets fed into the Calvin cycle in a different cell. But in CAM, it just happens different times of the day. So CO2 is put into this, the acids at night, and then they get released during the day and made into sugar. So wrapping back to the big picture, the energy that enters chloroplasts as sunlight gets stored as chemical energy in organic compounds. Sugar that is made in the chloroplasts supplies the chemical energy in carbon skeletons to make the organic molecules of cells. And then plants will store excess sugar in the form of starch in chloroplasts and other structures like the roots, tubers, seeds, and fruits. So again, this is the big picture of photosynthesis. And these are the learning objectives for this lecture. Okay, so that is it. That is lecture 8, photosynthesis.